Welcome back, everybody. Today we start our study of the cell cycle, cellular reproduction, mitosis, and then we'll extend that further into meiosis and sexual reproduction. So let's get started. When a cell divides, it needs to produce two identical daughter cells. If we were to just chop the cell in half, the two halves would get enough organelles, enough membranes, enough building block molecules to survive and grow. But the nucleus cannot simply be cut in half because each daughter cell must get identical copies of the DNA. So this means that cells must replicate their genetic material before they divide, and you've already seen how DNA replication occurs. And then that DNA must be carefully pulled apart so that each daughter cell gets a full copy of the genes. The process by which the DNA is separated is called mitosis. And we can imagine this entire process of cell division as occurring in a giant cycle. So actively growing cells go through a cyclic process that leads up to division into two daughter cells. Let's take a look. Here's a picture of the cell cycle, and I recommend you stop the video and put this diagram in your notes. Historically, the cell cycle has been thought of as an interphase punctuated by these moments when the DNA separates and the two cells then divide, called the mitotic phase. So interphase and mitosis. And mitosis itself can be broken down further into the separation of the DNA and the separation of the two daughter cells. Now, in recent years, let's say the last 40 years or so, interphase has been broken down into several other components, the so-called G1, S, and G2 phases. G1 literally stands for the first gap phase, G2 for the second gap phase, and S for the synthesis or the DNA synthesis phase. So let's take a look at these in a little more detail. In G1, the cell actually grows back to normal size. So the daughter cells are about half the size of the original cell. So now G1 is concerned with growing the cell back to normal size, creating new organelles, and the cell, of course, has to live during that time. Then in the S phase, DNA is replicated. If the DNA becomes replicated, then the cell is going to have to continue through mitosis and cell division. In G2, after DNA is replicated, the cell continues to grow. It checks the DNA to make sure it was copied correctly so that it tries to eliminate mutations and makes preparation for mitosis and cell division. This information shows us how mitosis fits into cell growth and reproduction. Now, the next thing we want to do to get us ready for understanding mitosis is to look at chromosomes and their structure in more detail than we have already. Eukaryotic chromosomes consist of a material called chromatin, and chromatin is actually a complex of DNA and proteins. Up to now, we've sort of considered that the DNA is just the DNA, but in eukaryotes, it's actually a complex with proteins. Almost all cells have two sets of chromosomes, and so what that means is that they have a pair of each chromosome. One set comes from the individual's mother and the other from the father. And these are called homologous chromosomes. So that's vocabulary you need to know. These homologous chromosomes contain homologous genes. In other words, genes on each chromosome that are the same gene, although not necessarily the same form of a gene. And if that doesn't make sense to you, we'll clarify that in the next unit. And these genes occur at the same physical location, which we call a locus, on the chromosome. At the start of cell division, the chromosomes condense. And what that allows us to do is actually see them. Otherwise, they're non-condensed. What this means is that in order to carry out transcription and translation, chromosomes need to be in their non-condensed form. And condensation occurs simply for the process of pulling the DNA apart. Let's take a look at this. In this electron micrograph, you can see in the middle a cell with condensed chromosomes, whereas these two cells have decondensed chromosomes where you can't see the individual chromosomes. This cell is preparing for cell division. These maybe preparing for it, but they're not going to do it imminently, whereas this cell is about to divide in the next 5 or so minutes. When the replicated chromosomes condense, they appear to be X-shaped. And so why is that? Because each side of the X is one complete chromosome. And I'm going to show you a picture of this in a second. The center of the X is called the centromere. One of the things you're noting is there's a lot of vocabulary today, and this is really important that you get this vocabulary down because we need to be able to use it immediately. And the centromere holds the replicated chromosomes together. Each side of the X is known as a chromatid, and together they are referred to as sister chromatids, which, by the way, would be a great name for a band. Hey man, remember when we saw Sister Chromatid down at the Michigan Theater? Yeah, wow, that was really cool. Yeah. If no mutations occurred, then the chromatids are identical. They have the exact same sequence of DNA nucleotides. So let's take a look at this. Here we can see the X shape I was talking about. And in a second, I'll show you an electron micrograph. And here are the sister chromatids right here. And this would be the centromere. Normally, when the cell is not in active division, the chromosomes will look like this, but they won't be visible. They'll be a single, long, linear strand of DNA. The other thing I want you to notice about this is that the form of the gene is different. It's called allele big A over here and allele little a over here. So on the replicated chromosome, the big A's are on the identical loci on the sister chromatids, and the little a's are in the same place on the sister chromatids. Here is a photo of a replicated chromosome, and you can see the sister chromatids are here and here. These are genetically identical except for mutations. What this shows you is the process that occurs during the cell cycle. At the beginning of the cell cycle, the chromosome is unreplicated. Then it becomes replicated and looks like an X, and we can actually see it. And then during mitosis, they get separated. And during cell division, which is referred to as cytokinesis, the two new cells form. Now, one of the reasons we know so much about chromosomes is because we can see them during mitosis. And one of the techniques that was used to study genetics in the very early days was to take photos of cells during mitosis, blow them up, and then cut them out of the photo and pair them up into what's called a karyotype. So when homologous chromosomes are paired and put together in a display such as this is called a karyotype. Even though the genes are too small to see here, there are banding patterns on the chromosomes, and scientists use that to gather information about inherited characteristics of an organism. So what do you notice about this karyotype? Maybe you pause the video and look at it for a second. Okay, hopefully you're back. So first thing is the chromosomes are paired. There's 2 of each. In this human karyotype, there are 23 pairs of homologous chromosomes for a total of 46 chromosomes altogether. You will notice also that one set of chromosomes is labeled with an X and a Y, and some of you may remember that these are the sex chromosomes. So the biological sex of the person who had this karyotype is male. And the other thing that a trained geneticist could see is that chromosome 18 is aberrant and is related to a series of disorders. Now, you and I can't tell that because we're not trained to recognize these chromosomes, but a trained geneticist could. So, to review what we've learned about chromosome structure, in preparation for cell division, DNA is replicated and the chromosomes condense. Each replicated chromosome has 2 sister chromatids that will separate during cell division. Each chromatid is an exact copy of the other, except for mutations, of course. And the centromere is the narrow waist of the replicated chromosome where the 2 chromatids are most closely attached to each other. In the next video, we're going to discuss the process of mitosis itself. And that will require you learning a lot of new vocabulary, but it's absolutely essential that you learn today's vocabulary in order to be ready for the next video. What I hope you understand today is that cells that are actively growing and dividing go through a cell cycle, that during part of interphase in that cell cycle, they are replicating the DNA, and that in another part, the DNA is getting separated and the daughter cells are being formed. And I also hope you now understand the details of the structure of a chromosome. Until next time, go outside, take a look at nature, ask biological questions, bring them back to class, and I'll see you soon.